the introduction. So as just said, I will be talking today about how microbes get to live together, how they form communities, how they get to reach this fantastic diversity unparalleled in any other groups. So the general agenda of the talk will be first a very brief introduction to microbial ecology and the problem of how organisms live together in general. Then I will be showing you three interesting results from three different projects on the topic of how microbes live together, and then a quick mention of what we are doing now and what the future research looks like in the group. So let's start with the introduction to microbial ecology. So before we actually go into it, I must warn you that methods will be very, very briefly discussed. Concepts will be very broad and terminology somewhat loose, and also biological context will be scarce. This is a bummer for any biologists in the room, but I feel it's necessary to be able to reach a wider audience. But with that say, towards the end, when we hold the discussion, please feel free to ask anything, whether it's a general question or a very specific question about methods. I'm happy to address that. So first of all, there are three big concepts in microbial ecology that are related to how microbes live together. The first one is the concept of community assembly. Community assembly is this it explores the set of rules of assembly, and by assembly I mean how organisms get to form communities, live together in the same place. It studies the effect, the relative contributions of the environment, the different organisms, migrations, and other factors in the community assembly, and also the role of stochasticity. How much of it is just driven by chance? There is a second related concept, which is succession theory. Succession theory studies the steps of succession, the order of colonization, and the effect of disturbance. This is probably the best known concept in ecology because it's very often the only major concept in ecology that is actually discussed in high school around the world. So some people have some idea of this, at least some intuition of this succession theory. And importantly, one part of it is the effect of disturbance, which we will be discussing in more detail. And then the third component of this problem is coexistence theory, which is really the mathematical framework that evaluates how is it possible that actually different species coexist in the same ecosystem, in the same environment, given a small number of limiting factors. So traditional ecological theory indicates that the number of species should be limited by the number of limiting factors, but we know now that the actual number of species that live in a given place is much higher than that, and it's even higher for microbes. So this coexistence theory is the mathematical formulation of that problem. But in fact, in this talk, I will be focusing on the first two concepts. I will not be discussing the mathematical principles of coexistence theory. Now, this is in general for ecology, for any group of living organisms. But there is a big problem with microbes, and that big problem is that they are small. This is here a cartoon of bacterial cells, just to give you an idea how these bacterial cells compare to a small eukaryotic cell. They are about 10 times smaller in diameter. A small eukaryotic cell, in turn, is about 10 times smaller than a large eukaryotic cell, again in diameter. And this is how a large eukaryotic cell compares to 1 square millimeter, which in turn, depending on the resolution of your screens, it's probably 1 or 2 orders of magnitude. This square is probably 1 or 2 orders of magnitude larger than an actual millimeter. So big problem, they're very, very tiny. As you can see here, there is a little tiny speck. That's not the bacteria. That little tiny speck that we can barely see is a small eukaryotic cell. We cannot even see here bacteria. So of course, this poses a big problem, and that problem is we cannot go to the field and directly observe them in the wild as we can with animals, as we can with plants, as we can with most of the basis organisms that have been used to develop ecological theory. However, there is one advantage. Many biologists will say, well, yeah, that's true. You cannot see them in the wild, but you can take them and multiply them and cultivate them, and you can end up with millions or billions of cells very, very quickly. This is called cultivation, and it's the process of extracting a given bacterial or archaeal cell or fungal, but I will be talking about bacteria mostly today, from an environment, isolating it, meaning extracting only one parent cell, and then propagating it in the lab, right? This is the process of cultivation. And the way it generally looks is we go to an environment, be it land, sea, or air. And yes, there are, we do microbiology of the air. There are communities, known communities in the clouds, for example. So we go to an environment, we take a sample, and then in that sample, we can actually either observe directly through microscopy or related techniques like FACS or different ways of counting cells. There are different techniques that we can use that are associated or related to microscopy, where we can actually observe directly from that sample and count the number of cells in there. Or we can take this sample and plate it into a petri dish or a broth, and then actually grow them. The big problem with that is that you can see using something called dilution techniques. This process here on the right, this process of cultivation, can actually result in a very, very accurate quantification of the number of cells that gave rise to the colonies here. So we can very accurately estimate from how many original cells we actually got descendants in our plate. And of course, from this method, we can directly count the number of cells in the sample. Problem is, they don't add up. Those two estimates are very, very, very different. The number of cells that we can actually see directly without cultivation, it's much, much larger, orders of magnitude larger than the number of cells that we can actually count. In fact, this effect is so general, so universal, and so prevalent and so strong that it gets a catchy name. It's called the Great Plate Count Anomaly. And what we have seen reproducibly in many different environments is that around 1% of bacteria in a sample can actually be cultivated. Meaning we have about 99%, it really depends on the environment, but about 99% of organisms that we have no way of actually growing in the lab. We cannot cultivate them. Those are the uncultivated, the jet uncultivated organisms, and it's the large majority. The thing is, there are other ways of studying microbes. We can bypass that problem. We can use environmental genomics, and environmental genomics offers an alternative to study the uncultivated majority, which can be this map here shows, or this stack bar plot shows the number of organisms for which that are somewhat related to something that we have cultivated, and these in white are the ones that are completely different from anything that we have cultivated. And all of these we can know directly from using environmental genomics, and in particular a technique called metagenomics. So metagenomics is the process of going to the field, sampling water or air or soil or whatever it is, and directly extracting the DNA and analyzing that DNA, as opposed to going through cultivation. So this bypasses entirely the process of cultivation. So now we can actually now study microbes using this kind of techniques. So this brings us to the first chapter of chapter 1. I cannot be everywhere at all times. So here you have three pictures of 3 organisms that probably everybody here is somewhat familiar with: a koala, a pine tree, and a coffee plant. Think for a moment where they live. Where do they live? Can you actually tell where they actually typically live? Can you pair these photos of these organisms, the koala, the pine tree, and the coffee tree, with locations around the world? And this is a map of the tropics, this is a map of Australia, and this is a map of the Northern Hemisphere. And I'm actually gonna ask you to answer this. I didn't tell you, but there was a quiz in this presentation. So just to remind you, this one here, the brown one is the tropics. This is a map of Australia, highlighting Eastern Australia. And this is a map highlighting the Northern Hemisphere. Okay, perfect. Not everybody has voted, but I have already a very clear picture. Everybody knows where these organisms come from, or where these organisms typically live. And the reason for that is that they have a very well-defined range, geographic range. The study of that geographic range in general is called biogeography. And biogeography is very clearly defined for large organisms like animals and plants. But let's talk now about biogeography for a moment, and I'm gonna make a very quick detour here. If anybody's into musical theater here, they might probably recognize this song, "Why Do We Build a Wall, My Children, from the famous musical Hadestown. In this song, what we have is this idea that something like building a wall can produce a domino effect that produces jobs, that produces that defeats poverty, that increases opportunities for everybody, and in turn that improves the lives of people in a given community. So it makes them different from their neighbors. But of course, there is a simpler explanation, and is that by building a wall, you separate yourself from your neighbors. So there is a direct effect. If you separate yourself from your neighbors, you're more going to be differentiated. That one problem of whether this separation and differentiation are a direct or an indirect causality is at the core of biogeography. So what we have is, instead of a wall, we have distance. We have actual geographical distance. Does the distance between organisms or between locations cause differentiation? That's called dispersal limitations. Or is the distance, because more distant locations have different environments, and different environments select for different organisms, is that what causes the differentiation? So are there dispersal limitations or environmental sorting? And in general, it's recognized that in most organisms, the biogeography is the result of both of them. Although in some cases, when we talk about biogeography, we mostly focus on dispersal limitations. But the thing is, at the very early in microbial ecology, even before the term microbial ecology existed, there was this phrase by the Dutch microbiologist Lawrence van Becking that has become a mantra of the field that says that everything is everywhere and the environment selects. So basically what Bas Becking is saying is that dispersal limitations don't even matter at all. There is no effect of dispersal limitation. Distance doesn't matter because microorganisms, because they're so tiny, they can disperse so easily. So they're basically everywhere around the world. And all that matters is environmental sorting. So that becomes an open question. Do we even have biogeography in microorganisms? So to answer that question, we went to the Chattahoochee River, which is a river that covers much of the southeastern continental USA, and has a series of 5 lakes and 2 estuarine locations along the riverbed. And just to give you some context, this is actually the distribution of land cover in the basin of that riverbed. And what you can see is that in general it's a very stable land cover. Sure, there are some, there is here, for example, the city of Atlanta that has a large urban cover that feeds into the second lake, West Point. And there is also some agricultural input feeding into the southernmost lake, the Seminole. But in general, we have a regularly stable land cover, with some exceptions that we can actually use leverage to identify statistically the effect of land cover on the communities. Now, I'm gonna completely bypass the methods and just go straight into the results. And what we see here is each one of these dots represents an entire community from one of the different lakes. So the lakes are here highlighted with these polygons. And what you can see is that communities sampled from the same lake are more similar to each other than communities of other lakes. This is an ordination plot where the distances in the plot somewhat reflect the actual differences between those communities. And just to give you an idea, well, first of all, 72% of the variance in this entire collection is explained by location, but of course that includes the effect of different environments. So there's, well into the south, we actually go into an estuary. So estuaries are very different from lakes. Of course, they are different. But just location, just the distance along the river already explains 40% of the variance. To give you an idea of how much 40%, how strong that is, the best known effect in freshwater communities is the effect of seasonality. Seasonality determines very strongly what kind of communities get to grow in a given lake. We know that lakes in general have this cyclic process, and they are very, very seasonal. And seasonality only explains 49% of the total variance. So that means that biogeography is almost as strong as seasonality, even though seasonality is the best known factor in community assembly that we know of in the lakes. Not only that, not only have we identified that, yes, indeed, biogeography distance itself is very strong, and we can actually statistically, as I mentioned, tease out the effect of other physicochemical parameters and the formation of an ecocline along the river, and things like land cover, and none of that is as strong as distance itself. And not only that, we can actually go and see which organisms grow where. Here, the pink boxes indicate different organisms in the different columns, and the different rows are different lakes. So we can see who is growing where, which communities are being formed, and not only that, we can actually identify something that is called provinces, microbial provinces, which are groups of lakes that actually form consistent communities. So kind of regions, natural regions that were inside which there is much more migration than between them. So now that we have identified the effect of biogeography, the next question is, okay, microbes are affected by the distance. How do they actually overcome that? What is important on overcoming that distance or on traveling farther? So I'm gonna actually have this takes us to the second chapter, and I'm gonna ask you to vote here on who can travel to more places. And bear in mind, this person can probably go higher up in the mountains, but this person might have anything from snorkeling equipment to a tuxedo. So it's not an obvious answer. Who do you think can go to more places? Wow, this is remarkable. We have exactly a 50-50 split. And you're actually, you're both correct. This is a really hard problem. I don't know the answer a priori. And this is at the core of a well-known trade-off in genome size, or a somewhat publicized trade-off in genome size in the field. And it's, on one hand, we have something called the Black Queen hypothesis. The Black Queen hypothesis tells us that smaller genomes are better for organisms, because the smaller the genome is, the faster organisms can grow, and also the less energy they spend doing functions and expressing functions that are not necessary in most cases. So on one hand, organisms have these, microorganisms, prokaryotes specifically, have this very strong incentive to have very tiny genomes. But on the other hand, the more genes they have, the more functions they can perform, the more diverse their metabolism is, the more environments and they can colonize. Right? So is this exact problem? Am I better off having a small backpack that I can take everywhere? Or am I better off taking a bunch of things, some of which I may or may not use, but I will be prepared for everything? We don't know. We don't know. And so we went out to test, we set out to test this question, to see what the answer was for this question. And we used actually a collection of genomes that we were able to recover from all across the tree of bacteria. So you might not be familiar with phylogenetic trees, but just to give you some context, this circle represents most major clades in known major clades in bacteria, and these longer blue lines represent all the genome representatives that we were able to extract from this region that we were talking about in the Chattahoochee Lakes. So we have this fantastic collection of all across the tree of bacteria, specifically from freshwater or from aquatic organisms, that we actually were able to use to taxonomically describe at least 3 different new taxa, 3 different new organisms. And we took this collection and we said, okay, now we know where they are in the region. Let's now check out where they are around the world. So we went to check, of course, the location. This here, the different rows are, each row is a genome or a different species. And each column in this matrix represents a different location. So here in the corner, we have the 100 samples from our lakes. And estuaries, the estuaries are here. So from our lakes, we have other lakes. We have a large collection from Lake Mendota in the US. We have lakes, bogs, rivers, estuaries, and marine samples from around the world. And well, one thing that we immediately noticed is that most of our organisms are actually restricted to aquatic environments. It doesn't matter, even groundwater is not a very good place to look for these organisms. So, okay, we have a clear aquatic, marine, and freshwater collection of organisms, but how far can they go, and how different environments and biomes can they actually thrive in? And what we observed is a significant, weak but very significant negative correlation between the expected genome size and the biome count or the habitat breadth. And this also extends to geodesic ranges, to latitudinal ranges, to a bunch of different metrics of distribution. So the answer was, those of you that thought that voted A, you were right. At least for this, in this case, for these microorganisms, they are better off having lighter backpacks, having smaller genomes. But something that was even more striking is that a much stronger effect was actually found for coding density, even stronger than the effect for genome size. So what we see is there is this trade-off between the Blackwing hypothesis and the increased habitat and geographic ranges. So one is pulling to have more genes, the other is pulling to have smaller genomes. And at the end, what you end up with is selecting for higher coding density, meaning that regardless of whether you have a small backpack or a large luggage, the most important is that you actually organize it well and pack in a way that you maximize the space in that backpack. That was completely unexpected. In retrospective, it's a natural expectation from this question, but that was unexpected to us, and it was an interesting result that we identified. And then all of this is assuming that we have a very constant environment, right? That we have an environment that doesn't change too much. We're assuming no adversities in this environment. But that's, of course, not always the case. So last question for today: who can survive better unexpected adverse situations? And here on the left, we have the specialist. For those of you that haven't seen that movie, Stallone plays an expert in explosives. He knows how to blow things up or how to prevent things from blowing up. And then on the right, you have MacGyver, who is not great at anything, but he's really good at everything. He's just good at doing any different tasks, even though he's not an expert in any one of them. And I don't have all the responses yet, but the picture is very clear. You all think that MacGyver can survive better unexpected adverse situations. And the key word here is unexpected, right? We don't know what to expect, so we cannot really prepare for one thing. The best we can do is maybe prepare somewhat okay for everything we can think of. And just this thing that you have just postulated with your voting is actually known as, in fancy ecological terms, as the specialization disturbance hypothesis, or SDH. Now, specialization, of course, is a metric of how narrow the specialty or the expertise of an organism is. More technically, what that means is a measurement of the niche breadth. So the narrower the niche breadth, the more specialist an organism is. So this is the specialist here in 0, and this is MacGyver here in 1. Disturbance is the technical term for an unexpected adversity. And what this hypothesis says is that the more generalist an organism is, the better it can survive or it can respond to disturbance, to an unexpected adversity. The problem with and this is well established in both animals and plants. The problem with microbes, though, is that we have this extreme diversity. Of course, that's a problem for us ecologists. For microbes, it's fantastic because they have so much diversity. They actually have way too many specialists. So if you think about this problem of having too much diversity for us ecologists, what we have here is the extreme diversity causes that you have specialists for pretty much anything. So if you have specialists for anything, there is no unexpected adversity. Nothing is unexpected because any one given adversity that you bring to the table will result in a selection of that one specialist. Some microbe at some point was expecting that. So that's a problem. That's an open problem for microbes. Does the specialization disturbance hypothesis actually apply to microbes, or does it break because of this extreme diversity? And there was a natural experiment, these catastrophic events of April 2010 that have been come to be known as the BP Deepwater Horizon oil spill, or in formal official documents as the Deepwater Horizon oil spill, because BP was able to take out the BP name from it. But we all know that the rig was owned by that company. So when this happened back in 2010 in the Gulf of Mexico, of course, this was one of the largest accidental environmental catastrophes in North America, or at least in U.S. American history ever, probably in human history, one of the worst, at least accidental. But what we were actually able to use this for was to study what happened when this giant catastrophe affects a community, a microbial community. And for that, our collaborators at Florida State University actually rushed to the beach and were able to collect sand samples before the oil reached the shoreline. So these are samples, these green ones are samples before. Then they kept taking samples during the oiling and after, up to a year later. These are the oil samples, and these are here the samples a year later. And what, again, these plots here are ordination plots. The distance in the plot, it somewhat reflects the difference between these communities. And what we can see is that indeed there was a strong effect both in the makeup, in the taxonomic makeup of these organisms, who was there, but also in the functional makeup. What were they doing? So these were very strong community composition shifts. One thing that was highly expected is that the effect of that catastrophe will be to decrease taxonomy. And that's exactly what taxonomic diversity. So a catastrophe came, many of these organisms just died because of the toxicity of the oil, or because they were unable to gather enough nitrogen, or because of anoxia. But for many different reasons, the diversity actually will decrease. So we had a significant effect and a significant decrease in taxonomic diversity when the oil reached the shoreline, and then it came back afterwards, right? So in the year after that, that diversity was recovered. So this is similar to, if you think a group of travelers, we have this large group of travelers, and then there is this catastrophe, this unexpected catastrophe. Before, we had a high diversity of nationalities represented in our group, and then the resulting group now has a very small diversity of nationalities. But if that is the case, and it doesn't really matter what how many objects these travelers had, or how many different functions these organisms performed, we should expect the exact same on functional diversity. But we actually observed the exact opposite. There was an increase in functional diversity during the oiled samples during the oil, the effects of oil sands, and then decrease down a year later. So basically, that's like saying we took instead of taking the passports of these passengers, we just took the objects in their luggage and classified it and measured the diversity. And what we see is that before this catastrophe, there were very few different objects. There was a low diversity of objects. And after the catastrophe, there are fewer nationalities, but a higher diversity of objects. So what this is, what we interpret this as, is as evidence that indeed, being a generalist, being MacGyver, actually helps survive better an unexpected adversity, which is exactly what you postulated with your vote. Now, the last problem with this result, it was beautiful. We were able to show this very clearly. Decrease in taxonomic diversity, increase in functional diversity. This was published back in 2015. But the problem is that we still had this open field. So the fact that we saw it in the environment, in the wild, was great for many reasons, but it also posed some challenges. We had variable weather, migrations, seasonality, a bunch of confounding factors, and we didn't know if this would be important or not. So what we set up to do was we reproduced the entire experiment, but in the laboratory. And this was published last month in the ISME journal. We actually observed, we got a confirmation of this. We observed the exact same pattern in the laboratory that we had observed in the open field. So that's the answer. MacGyver actually can survive better unexpected adversity. Also, even in microbes, where we didn't know for sure that was the case. And there are different publications now. That there is one other publication that actually came out in the same issue of that same journal earlier this month, where they have a completely independent confirmation of that exact same observation. Then now to conclude here, I just wanted to tell you very briefly what else we're doing. It seems like we've been answering different questions on how microbes get to live together and what matters for their, what kind of genomic features matter for their survival. We're extending to other environments. We're working in the halophile sequencing project. We're officially part of the halophile sequencing project led by Professor Ramon Rosselló Mora in the Balearic Islands, where we are actually studying these same problems in salterns all around the world, anywhere from Argentina to Israel. The second problem we're trying to address is the problem of who's there. I have been telling you who's present, who's absent in those visual representations of matrices, but that's actually a problem that is not solved. We don't really have a very strong, very robust statistical framework to define detection. So we're working on it in the lab, and we're also working on the problem of classification and nomenclature, how to classify and name these organisms that we cannot grow in the lab, but that we know exist because we have their genomes. And then finally, we're also working on problems of definitional challenges and integrative statistical approaches to put together all these different concepts into common frameworks. And with that, I would like to thank all the different collaborators that played pivotal role in all these different projects that I have been showing you today, and of course the funding agencies that made them possible. Thank you very much.
And I